Om Swastiastu. Thank you for the opportunity. In this question, I will presenting about biochemistry middle exam. My name is Ana Agung Ayu Putri Adiani with SIN 2413031004. Okay, the first one, the topic is cell membranes and bioenergetic. This is a case. An experimental drug significantly increased mitochondria membrane fluidity, but it also caused proton or hapless to leak across the membrane. And the question, evaluate the drug's effect on the proton gradient, oxidative phosphorylation efficiency, and ATP production. Design a strategy for modifying the drug molecules to maintain its therapeutic effect without disrupting cellular energy homeostasis. Okay, the answer, under normal condition, the electron transport chain generates a proton gradient that is utilized by the enzyme ATP synthase to produce ATP. However, certain drugs increase membrane fluidity, making the membrane more permeable to protons. As a result, protons return to the matrix without passing through ATP synthase. Proton leakage occurs, causing the proton gradient to decrease, disrupting chemiosmosis and reducing ATP production. This condition causes an imbalance between oxygen consumption and ATP production, which can lead to an energy deficit and even apoptosis or necrosis in several cases. Okay, uh, the strategy for modifying the drug molecule to maintain its therapeutic effect are restricting molecular mobility within the membrane through a more rigid structure. The second one, using a prodrug that is active only at the target site to minimize effect on healthy mitochondria. The third, reducing proton-carrying properties or protonophilic through functional group modification. And the last one is specifically targeting membrane lipids to avoid interfering with electron transport chain proteins. Okay, we will move to the next question. That is about metabolic engineer and alternative energy. The case, a team of scientists aim to engineer human cells so that they can efficiently use acetate as their primary energy source under conditions of extreme glucose deprivation. Design a metabolic engineering strategy to achieve this, including the enzymatic pathways that need to be enhanced or inhibited. Evaluate the potential metabolic risks. The answer is acetate in the body can be converted into acetyl CoA and enter the TCA cycle to produce NADH or FADH2, which are used in the electron transport chain to generate ATP. However, its natural utilization is limited. To enhance this utilization, metabolic engineering is employed to optimize the conversion of acetate to acetyl CoA via the enzyme acetyl CoA synthase or ACSS2. This is achieved by overexpressing the ACSS2 gene and targeting it to the mitochondria to increase the substrate supply through the TCA cycle. Next, the activity of rate-limiting enzyme in the TCA cycle was increased to maintain NAD+ balance, thereby ensuring that metabolic processes continued to proceed optimally. Modifications were also made to the electron transport chain proteins complex to enhance ATP production efficiency and maintain mitochondrial membrane stability. However, this approach carries risks such as metabolic acidosis and excess of electrons that can damage proteins, the accumulation of acetyl CoA, and an energy imbalance within the cell. And this is the cycle of the glycolysis. fatty acid and the acetyl CoA. Okay, we move to the next question. Uh, there is a case in patient with a severe G6PD deficiency. Conventional antioxidant therapy is less effective. And the question, evaluate the limitation of this therapy and design a molecular or genetic-based therapeutic approach to enhance erythrocyte resistance to oxidative stress. Okay, so the in uh, G6PD deficiency, conventional antioxidant therapy is less effective because it does not address the underlying cause, namely low NADPH production due to a disruption in the pentosaphosphate pathway. NADPH is required to maintain glutathione in its reduced form GSH, which protects red blood cells from oxidative stress. Antioxidants such as vitamin C and E only temporarily scavenge free radicals and cannot regenerate GSH without NADPH, leaving red blood cells vulnerable to damage and hemolysis. This limitation is exacerbated by the fact that red blood cells lack a nucleus and organelles, making them unable to increase the production of antioxidant enzyme. As a solution, gene therapy can be formed by introducing the normal G6PD gene into hematopoietic stem cells so that the resulting red blood cell can produce an ADPH optimally. Other approaches include the use of small molecules to active and stabilize the remaining G6PD enzyme, as well as the administration of glutathione precursor or NADPH analogs to enhance the cell's antioxidant capacity. Okay, we'll move the next question with the topic glycolysis and cancer, the Warburg effect. The question, cancer, sorry, cancer cell exhibit increased glycolysis even in the presence of oxygen, or the Warburg effect. And the question, evaluate the metabolic benefit of this strategy for cancer cell. Develop a therapeutic approach based on inhibiting the glycolytic pathway without significantly damaging normal cell. Okay, so cancer cell exhibit the Warburg effect, meaning they will rely more heavily on glycolysis even when oxygen is available. This strategy offers advantages such as rapid ATP production, the provision of intermediates for biosynthesis, which is nucleotides, amino acid, and lipid, and the creation of an acidic environment from lactate that supports infection and suppresses the immune system. Additionally, reduced reliance on mitochondria helps minimize the formation of ROS, which can trigger apoptosis. This phenomenon is also utilized in cancer therapy by targeting K-glycolysis enzymes that are overexpressed in cancer cells, such as um, hexokinase 2, pyruvate kinase, and the glucose transport. Inhibiting this target can disrupt the energy supply and biosynthetic processes of cancer cells. To enhance uh, specificity, the therapy can be combined with delivery systems such as nanoparticles or prodrugs that become active within the tumor microenvironment, thereby effective targeting cancer cell without damaging normal cell. Okay, now we move to the next topic, gluconeogenesis and insulin resistance. There is a case. A new drug has been developed to suppress hepatic gluconeogenesis in patients with type 2 diabetes, but it causes hypoglycemia in some patients. And the question, evaluate the causes of the side effect and develop a regulatory strategy that specifically targets the gluconeogenesis pathway without disrupting normal glucose homeostasis. In type 2 diabetes mellitus, drugs that suppress hepatic gluconeogenesis can cause hypoglycemia by excessively inhibiting glucose production without taking the body's condition into account. In fact, gluconeogenesis is essential for maintaining fasting glucose levels through enzymes such as PAPCK and glucose 6-phosphate. Non-specific inhibition also disrupts homeostasis because it does not distinguish between hyperglycemia and normal condition, and ignore metabolic regulation such as the AMPK system and energy balance. As a solution, therapy needs to be made more controlled, for example by targeting upstream signal such as glucagon receptor or inhibiting gluconeogenesis only when glucose levels are high. Other approaches include the use of prodrugs that become active during hyperglycemia, moderate activation of AMPK to balance metabolism, and RNA-based therapy. The partially reduce enzyme expression without completely halting glucose production. Okay, now we move to the next question with the topic the Krebs cycle and mitochondrial disease. Uh, there is a case a patient has a partial mutation in the alpha ketoglutarate dehydrogenase enzyme. Evaluate the impact of this mutation on metabolic flux in the Krebs cycle and related pathways. Design nutritional or pharmacological intervention to mitigate the effect of energy deficiency. Okay. Partial mutation in the alpha-ketoglutarate dehydrogenase enzyme inhibit the conversion of alpha-ketoglutarate to succinyl CoA in the Krebs cycle, thereby reducing the production of NADH and ATP. This leads to the accumulation of alpha-ketoglutarate, disruption of metabolic flux, and subsequent effects on amino acid metabolism, nitrogen balance, and increased oxidative stress. To address this nutritional and pharmacological intervention are implemented, such as the administration of alternative substrate, uh, such as succinate and medium-chain fatty acid, vitamin B1, supplementation as an enzyme cofactor, the use of antioxidants, and the enhancement of electron transport chain efficiency. Uh, Additionally, activation of anaplerotic pathway, such as via pyruvate carboxylase, help maintain the continuity of the Krebs cycle. This combined approach aims to minimize energy deficit and maintain metabolic balance. Okay, now we move to the next topic, the extreme metabolic integration, starvation versus ketosis. The case, in a state of chronic starvation, the body relies on ketogenesis. However, in some individuals, several ketoacidosis occurs. The question, evaluate the factors causing a shift from adaptive ketosis to a pathological state. Develop a prevention strategy based on metabolic and hormonal regulation. In chronic starvation, the body adapts by switching the ketogenesis, producing ketone bodies, and as an alternative energy source. However, under certain conditions, this can progress to ketoacidosis due to excessive ketone body production, which lower blood pH. This is triggered by low insulin levels and increased counter-regulatory hormones, leading to excessive lipolysis, elevated acetyl-CoA, and limitation in the Krebs cycle, thereby increasing ketogenesis. Other factors such as dehydration, kidney dysfunction, and diabetes also exacerbate the condition. Normal ketosis is actually well regulated, but regulatory failure or particularly insulin deficiency leads to uncontrolled ketone production. Prevention involves maintaining insulin levels and nutritional intake, ensuring adequate hydration, inhibiting lipolysis, and providing controlled amounts of carbohydrates to increase oxaloacetate, thereby directing acetyl-CoA into the Krebs cycle rather than into excessive ketogenesis. Okay, the next topic is about photosynthesis, optimizing crop yields. Um, there is a case, C3 plant super yields losses due to high photorespiration is in hot environments. The question, design a biotechnological approach to minimize photorespiration, such as through Rubisco engineering or alternative pathways, evaluate the advantage and limitation of this approach. Okay, in C3 plants, high temperature increase photorespiration because rubisco bind oxygen more frequently than carbon dioxide, thereby reducing photosynthesis efficiency and crop yield. Efforts to address this include engineering rubisco to be more selective toward carbon dioxide, utilizing more energy-efficient photorespiratory bypass pathway and increasing carbon dioxide concentration around the enzyme. The success of the strategies depend on proper metabolic regulation to avoid disrupting cellular balance. And there is a image that is the cyclus about Rubisco, this photorespiration, and this Kelvin cycle. Okay, the next topic is enzyme regulation and drug design. The case is a new allosteric inhibitor molecule was designed to inhibit PFK1, but it caused side effect in muscle tissue. Evaluate why this inhibitor has systemic effect. Develop a drug design strategy that is more selective for specific target tissues. Okay. PFK1 inhibitors can cause systemic effect because this enzyme plays a key role in glycolysis and is present in many tissues, including muscle. As a result, inhibition can reduce ATP production and lead to energy-related issues such as muscle fatigue. To enhance selectivity, strategies such as targeting specific isoforms, using prodrugs or nanoparticles, and designing inhibitors that are active only under certain conditions can be employed, thereby making the therapeutic effect more targeted and reducing the bedside effect. Okay, next. The topic is mitochondrial dysfunction and future therapies. Patients with mitochondrial disease inhibit multi-organ dysfunction. Current treatment is purely supportive. Develop innovative biotechnology-based therapeutic approaches such as gene therapy, mitochondrial transfer, or metabolic modulation. Evaluate their ethical implementation, effectiveness, and implementation challenges. Okay, in mitochondrial disease, impaired oxidative phosphorylation leads to reduced ATP production and multi-organ dysfunction. Treatment options include gene therapy or correct mtDNA, healthy mitochondrial transfer, and metabolic modulation, such as enhanced glycolysis or GOQ supplementation. Gene therapy holds the greatest potential for a cure but is still hampered by technical and ethical challenges, whereas other approaches are primarily supportive. Therefore, the best strategy is combination of gene therapy and metabolic support, taking safety and regulatory consideration into account. Okay, that are my presentation about biochemical. I'm so sorry if there's any mistake that I do in I presenting this material. Thank you for your attention to watch this video. I close it with Paramasanti. Om Santi 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 Om.